You will need a water bath, a clamp, petri dish, a pipette, ethanol, iodine, forceps, water, test tubes in a test tube holder, and you will need plants that will receive 3 different treatments. Photosynthesis is an important biochemical process in which plants, green plants, algae, and some microorganisms are able to harness energy from the sun together with carbon dioxide and water to produce glucose and give off oxygen as a byproduct. This is an extremely important process because these plants are primary producers or autotrophs, and they are where we obtain our food from. In order for us to better understand the process of photosynthesis, we need to understand all of the steps that take place during photosynthesis, specifically what the requirements of photosynthesis are. In this practical, we will be investigating the necessity of certain of the requirements of photosynthesis, specifically if sunlight is an important requirement, if the presence of carbon dioxide is a requirement, and lastly, if chlorophyll, which is a pigment found in plant cells, is a requirement for photosynthesis. In order to do that, we will be using an iodine test for starch. Iodine is a chemical compound that reacts with starch molecules and produces a very specific color change. Now, starch is several glucose molecules that are stored in plant cells. Okay, so if a plant is actively photosynthesizing, then there will be starch present in those plant cells. And if you drop iodine on a leaf that is photosynthesizing, you will observe a color change from a rusty orange color to a bluish black color. For today's practical, we have 3 treatments. The first is the dark treatment. This is to test whether light is a requirement for photosynthesis. In this treatment, half of the leaf has been covered with foil to prevent light from penetrating the leaf, and the other half has been left exposed to light. What we expect here, or the hypothesis for this treatment, is that the part of the leaf that was left uncovered will be photosynthesizing, and therefore, in the presence of iodine, will turn bluish black. The next treatment in the experiment is where carbon dioxide has been removed. Okay, so for this treatment, the plant was kept in a closed cupboard with a potassium hydroxide solution. This potassium hydroxide solution removes carbon dioxide from the air, and so the leaves of this plant, if carbon dioxide is a requirement for photosynthesis, would be unable to photosynthesize. In this instance, if you expose leaves from this treatment plant to iodine, you would expect no color change. Okay, so the iodine will remain a rusty orange color. The final treatment is a variegated leaf. Now, a variegated leaf is a leaf in which there is differential distribution of chlorophyll, the green pigment found in leaves, within the leaf. So you have some areas of the leaf that do not have any chlorophyll and other areas that do. If chlorophyll is a requirement for photosynthesis, then we expect that in the presence of iodine, only parts of the leaf that contain chlorophyll will actually turn a bluish black color, because that is the actively photosynthesizing part of the leaf, and the parts of the leaf without chlorophyll will remain a rusty orange color. Okay, so firstly, the first step that we will do is to remove a leaf from one of the treatment plants. In this instance, it is the dark treatment to test the effect of light, or the necessity or requirement of light for photosynthesis. So half of this leaf has been covered to prevent light from penetrating into the leaf. The first step is to place this leaf into a boiling tube. And then we fill this tube with water. The reason that we are boiling the leaf in water is to break down the cuticle and the cell walls, and we will use forceps to make sure that the leaf is immersed in the water, and then we will boil this leaf in water for 3 minutes. So the leaf has been boiling for 3 minutes in water, and we can already observe that the water inside the boiling tube has changed color slightly, which is a good indication that we have broken down the cuticle and the cell walls. So what we will do now is we will decant this water into a waste beaker, and we will put in some ethanol and boil the leaf again. The purpose of boiling the leaf now in ethanol is to extract the chlorophyll pigment. Okay? This is so that when we place the iodine solution on the leaf, we can better observe any color changes that will occur. Okay? Because the chlorophyll pigment is dark and will prevent us from clearly observing those color changes. So now I'm going to pour in some ethanol into this boiling tube, and we will again boil the leaf for 3 minutes and allow the chlorophyll to be extracted. So carefully place the boiling tube with ethanol back into this beaker with water and leave it for 3 minutes. So the leaf has been boiling in ethanol for 3 minutes, and we will now remove it. As we can see, the ethanol solution has turned dark green in color, which is a good indicator that we have extracted the chlorophyll as intended. Okay, so the purpose of that was to remove the chlorophyll so that we can observe the changes that we expect to occur in the presence of the iodine solution. So what I'm going to do now is to just decant the alcohol, the ethanol, and then I'm going to place the leaf on a petri dish. Remember, the ethanol has slightly dehydrated the leaf, so you need to very carefully open up the leaf again. The leaf is now a bit brittle, so you must be careful when unraveling the leaf. And what you now want to do is to just add a few drops of water to rehydrate the leaf. Okay, the leaf is now ready for the iodine solution. Okay, so this is the iodine. Remember that iodine changes color in the presence of starch. Okay, so if you have photosynthesis occurring, starch will be produced, and so iodine will change from a rusty orange color to a bluish black color. Okay, if photosynthesis is not occurring, we do not expect any accumulation of starch, and therefore the leaf will remain an orangey rusty color if there's no photosynthesis taking place. So using this pipette, we're just going to drop some of the iodine solution over our leaf. Okay, and we will let that develop for 2 minutes and then observe any color changes. Okay, so the iodine has been placed on the leaf, and we allowed that to develop for 2 minutes, and now we can observe the color changes. So just a reminder, this is a leaf from the dark treatment, or the treatment used to investigate the requirement of light for photosynthesis. So half of this leaf was covered with foil and the other half was left exposed to light. As we can see, half of the leaf remained a rusty orange color after iodine was added, and so we can conclude that no photosynthesis was occurring on this half of the leaf. On this half of the leaf that was exposed to light, the iodine has actually changed to a bluish black color, which indicates the presence of starch and therefore the presence of photosynthesis. Okay, so photosynthesis did occur on this half of the leaf.